Välkomna då till dagens föreläsning i Forskning i Framkant. Jag heter Håkan Hamberg och är professor i infektionsmedicin, och jag har nöjet att introducera Colm Nestor som är forskare vid Linköpings universitet inom molekylärbiologi och särskilt genetik. Och du ska prata om kan epigenetik förklara varför vanliga sjukdomar ökar. Välkommen.
Tack Håkan. And thanks to all of you for coming as well today. And as you can see from the title of my talk, I'm going to talk about if epigenetics can explain the massive explosion we've seen in common diseases in the Western world over the last 5 decades. And before I go any further, I'll answer that question by saying no, I can't explain it. But certainly it can maybe help us explain some of the increase we've seen in common disorders. So before we go any further, I'll explain what I mean when I say a common disease. Us geneticists like to classify things, and we classify some diseases as being simple. If they have a simple genetic cause, a single genetic cause that leads to disease like cystic fibrosis or Huntington's disease. However, many of the diseases we're used of and know, diabetes and rheumatoid arthritis and asthma and allergy, these are complex diseases, because you can't explain them by genes alone. Certainly, the genes you inherit can increase your risk of getting one of these diseases, but your environment and your lifestyle can also affect it. So regardless of what genes you have inherited, if you smoke, you increase your risk of getting lung cancer. Okay? So this is what we mean by a common disease, basically a very complex disease. And these common diseases are placing a huge burden on our healthcare services. In America alone last year, allergy, hay fever, cost the American taxpayer $500 million in healthcare. That's just for a very mild disease. Okay, so when you take them all into account, these common immune disorders are costing healthcare across the globe increasing amounts of money, and we don't really know what's causing them. We don't know what's causing their increase, and because of that, we can't effectively treat them with knowledge-based or intelligence-based medicine. We're just trying what's out there at the moment. So here's the good news. Here's a graph. It's 15 years old now, but it's a very famous one, and it's showing what we call primary infections. It's no surprise to people here in Sweden that things like TB and hepatitis and measles and mumps, they're decreasing massively in their frequency over the last 50 years. Polio and smallpox, these things are nearly eradicated worldwide. If we look over here, over the same time period, we see a huge increase in things like multiple sclerosis, diabetes, asthma, Crohn's disease, celiac disease. And in there also is allergy. In fact, allergy is the one that's shown the most significant increase across Europe of the complex immune diseases over the last 50 years. And this, trying to understand what's causing this is a major focus of all medical research out there at the moment. To understand how our environment and how epigenetics can affect our health and these diseases, we need to understand the genetics first. And who are we? Where do we come from? What makes us a human? Okay, getting down to basics. Well, we are the genes, we're the genetic material that we inherited from our parents. Okay, and these genes determine our height, our skin color, our eye color, our hair color. They determine lots about us. They also determine what risks we have from a genetic point of view for certain complex diseases. But we are also products of our environment. Like I said, if you smoke, where you're raised, your diet, it all affects your risk of getting diseases, your health, and your opportunities, both biological and social, in life. So we really need to take both a genetic and an environmental approach to understanding complex molecular diseases. So, right back to basics. This is possibly the most important and famous photograph ever taken in scientific history. And this is a photograph taken in an X-ray crystallographer of DNA for the first time. This was taken in 1953 by a fantastic lady called Rosalind Franklin. And being a boring scientist, she labeled it photo 51. The other 50 didn't work. And unfortunately, Rosalind Franklin died before the awarding of the Nobel Prize for the DNA structure. These two guys, Francis Crick and James Watson, used Rosalind Franklin's photo to figure out that DNA kind of looked like this, a spiral, a double helix. If you imagine taking a ladder by both ends and twisting it, that double spiral you'd get, that's a double helix. And here's a nicer photograph of it. We don't need to go too much into the structure of DNA, but DNA is basically made up of chemical letters, and there's only 4 of them. And our entire genome is made up of an alphabet of 4 chemical letters, A, T, C, and G. Now, it might be a small alphabet, but it's a very big instruction manual. There's 3.1 billion of these letters in our genome. And when I, as a geneticist, am working with it, I literally work it like this. I look at these sequences of the DNA. To me, it may as well be Japanese or Chinese. I can't read it. But to our cells, it's a very clear instruction manual of how the cells are to behave. So quite rightly, 15 years ago, when the human genome sequence was published, it was a huge feat. And it was rightly celebrated. I was working for Decode Genetics in Iceland at the time, and we had played a large role in this. So we were very excited about all the amazing information we were going to take out of the human genome in understanding what causes diseases and ultimately in developing new cures for diseases. It took 15 years to sequence that genome. It cost nearly $3 billion. But I said the human genome is a fairly impressive genome. It's 3.1 billion of these chemical letters, these bases. Coming along to now, we're nearly up to 10,000 genomes sequenced. It's only $1,000 to sequence an entire genome, and it takes around 2 days. And you can take a swab from your mouth, put it in the post today, and if you send them the money, they'll have it sequenced for you in 2 weeks. So we've come a long way. But quite rightly, at the same time, geneticists like myself knew there was an awful lot more to the story, and that we weren't going to get all the answers from the genome, despite the fact that that's how we publicized it. And how do we know there was something missing? Well, it's quite simple. We're all very diverse, all very different, all very beautiful in our own way, but once upon a time, everyone in this room looked exactly the same. We all were a single cell. And that single cell had one copy of the DNA that it makes up your body. And it divided and divided and divided. And it made up all the different types of cells in your body. These are incredibly different cells. And yet every single cell has essentially the exact same DNA sequence, the exact same blueprint. So how do cells control which parts of this DNA they're supposed to read? How does a kidney cell know how to turn on the kidney genes and not the brain genes? Why aren't our cells continually changing into something else? And this control is the field of epigenetics. And the word epi comes from the Greek above. So it's epigenetics is the layer of genetics above the DNA that controls the way our cells interpret DNA. How do we define epigenetics? Well, the most common definition is that any change in your gene's activity, okay, a gene being on or off, that is not caused by a change in the DNA sequence itself, must be epigenetics. There has to be something controlling the cell in telling it how to turn on or turn off that gene. But just like DNA isn't a scientific field, it's a molecule, epigenetics isn't itself a scientific field. So if someone tells you, I study epigenetics, you have to really grill them, because that's not enough. It's too broad. What exactly do you study within epigenetics? This is a simple diagram that shows you, for those of you not in the field, how huge epigenetics has become as a research topic across the world. This is a number of articles published every year about epigenetics since 1950. We're up to around 4,000 research articles a year on epigenetics at the moment. In the last 3 years, there's been more research articles published on epigenetics than there was on epigenetics in the entire history of human endeavor. So it's an incredibly popular, very hot topic. And hopefully I'll convince you that it warrants that it is right that we're studying it, because it's very interesting and important to our health. What does it look like? I'm not going to go into the molecular details of epigenetics too much here, but we can summarize epigenetics as happening in 2 ways. Our DNA isn't just a big long string of spaghetti, but it's actually not a bad idea to think of it like that. When you have spaghetti in a pot, and you put a spoon or a fork into that spaghetti, you usually take up the whole lot in one go, or you just get up one little string of spaghetti. And in some way, our DNA is organized like that within the genome. Lots of it are simply wound really, really tightly together. So tightly together that all the proteins in the cell can't physically get to the genes there. They can't get to those genes to turn them on. Other parts of our DNA have little chemical signposts on them called methyl groups, which basically say, don't come near this gene. Do not turn on this gene. And the way in which our DNA is wound around itself and where these little epigenetic signals are is different from each cell type. So kidney cells have a certain pattern of these, brain cells have a certain pattern, skin cells have a certain pattern of these. And it is in this way that our cells interpret our genome and know which gene should be on in which cell and at which time point. Methylation is not the most important one. There's no such thing as a most important type of epigenetic regulation, but it's the one I study. So it's my favorite, and that's what we're going to focus on today. And it's also the simplest one. This is what DNA actually looks like. We see this lovely double helix, this twirling structure. And this is two methyl groups. Now, if this was a gene, the proteins that want to turn on that gene in your cell simply can't get there because this is in the way. It's just acting as a hindrance. For some reason, unbeknownst to me, when DNA methylation was first discovered, they decided to start using lollipops to represent it. So you'll see a lot of figures like this, with white lollipops and black lollipops. And a white lollipop means there's no methylation. There's none of these chemical signals that say off. Black means there's methylation. Turn the gene off. So we can see some genes are unmethylated and turned on. Some genes are methylated and turned off. And there are proteins in the cell that can turn a gene from being unmethylated to methylated and methylated to unmethylated. So we can change these patterns in our cells if we need to. A very important thing, and one of the things that has really heightened interest in epigenetics, is that we can change it. If you have a mutation in the gene, we can't change that at present. We can't go into your cells that have the cystic fibrosis mutation and change it back. Maybe in 50 years we can do it, but we can't do it now. Epigenetics, however, we can change easily. There's lots of drugs out there that can change the patterns of epigenetics in your cells. Even things we eat, vitamin C, vitamin B12, folic acid. Exercise can change methylation patterns in some of your fat cells. Stress can rapidly change methylation at some genes in your brain. As I said, we even use drugs to get rid of methylation in some cancers. Smoking affects the methylation in your lung cells, even before you get cancer. So, the take-home message from this is epigenetics is changeable and we can control it to some extent. Next, this looks very complicated, but it's very simple. If we start with one cell that has a particular pattern, this gene is methylated and this gene is not methylated. As this cell divides into two cells, it keeps its methylation pattern. And as it divides into two more cells, it keeps its methylation pattern. Okay? And all of these cells together can make up, say, a liver or a brain or a skin. Okay? So, but this seems simple, but it's very, very important. It means that these epigenetic patterns are inherited. Okay? They're maintained somehow. And that is very important. If you take these two statements together, that epigenetic patterns can be inherited and epigenetic patterns can be modified by our environment or our experiences, you can possibly come to this conclusion, that our experiences can be transmitted to the next generation through epigenetics. We do something that changes the epigenetic pattern in our sperm or eggs, that is then translated to the next, or transmitted to the next generation. A lot of people like this. I like to assume this. It's a very simple and very appealing conclusion to these 2 points. It is not, unfortunately, true. The reality is some experiences may possibly be transmitted via epigenetics to the next generation in some species. But the reality is, in fact, that this transgenerational inheritance via epigenetics, there's virtually no evidence of this occurring in humans at all. In fact, there's very little evidence of this occurring in any mammals of any type. How it happens is also very difficult to understand. It is easy to understand if you smoke, how that smoke can affect the epigenetics of your lung cells, but exactly how it's going to affect the epigenetics of eggs in the ovary is not as clear. The best evidence for inheriting epigenetic changes, well, it's still just in plants and in worms sometimes that we use in research in the lab. So beware of quackery. Now in English, quackery just means people trying to sell you false cures. There's a big industry around epigenetics about how you can control your genes and the health of your genes that you pass to the next generation by somehow controlling your experiences in your current life in terms of diet, exercise, and even the way you think and feel and your beliefs. This appeals to a lot of people who don't like the deterministic idea of evolution, how it's just a bit random. People like the idea that I'm in complete control of my own biology, not only my own biology, but the biology I pass to generation after generation after generation. But the ready truth is, in humans, there's very little evidence of this to support this at all. One of the studies that really heightened everybody's belief and interest in epigenetics and epigenetic inheritance actually originates here in Sweden, in a place called Överkalix, which is way up in Norrbotten. Now, Överkalix is quite unique. Around 100 years ago, if you had a bad harvest in Överkalix, you were going to starve over the winter, because there was no way back then to get food from the south of the Sweden up to the top of Norrbotten in a bad winter. And there's lots of recorded famines, well-recorded famines in this county council between 1890 and 1915. So some very interesting and clever scientists said, well, why don't we go back and look at the people who were just children at this time, but who experienced either famines or times of plenty. And then let's look at their children and their grandchildren and see, do we see that there's been any effect? Because if you can inherit through some sort of epigenetic mechanism, maybe we think we'd see it here. Maybe the people who went through famine passed on something to the children and grandchildren that those who didn't experience famine didn't. What they found was, in the children of parents who had experienced a famine before, this is when they were young, pre-puberty, their children had a lower risk of cardiovascular disease, and their grandchildren had a lower risk of diabetes. So on the face of it, this study would say, well, yeah, there's something to this. And one proposed mechanism is that, okay, you have a normal pattern, whatever normal is, of methylation to start with. The people who went through the famine ended up with a certain famine methylation pattern, which they passed on to their children, and they passed on to their children after them. Whereas the people who had plenty to eat had a different pattern and passed that on. A very appealing idea. But there's absolutely no direct evidence in this study that it was through epigenetics. Okay? So how else could you pass this on? Well, it's actually very simple. Familial interaction. These children are being brought up by the parents who experienced the famine. Now, luckily, I have never experienced a famine, but I'm pretty sure if I did, I'd probably have a very different attitude towards food, what I eat, when I eat, whether I waste food. So it's very hard to separate, was this an epigenetic effect, or was it just the effect of growing up with parents who have had very traumatic but very different experiences? And this is why it's so difficult to study epigenetics in humans. It takes a very long time if you want to study grandparents, parents, and their grandchildren. And also, it's very hard to separate out the effect. Well, what is the effect of the epigenetics? And what is the effect of how they were raised by these people? So studies in animals are much more powerful. Okay, so we can control for a lot of these difficult and intangible effects on epigenetics. So here again at the University of Linköping, there's an excellent researcher called Per Jensen who in a study on chickens, and chickens are actually quite a useful animal to study, particularly for this reason here. Chickens are really, really resourceful. You can do something to a chicken, you can make it upset, or you can stress it out, or you can starve it. You can take the egg from that chicken, take it away, put it under the light somewhere else, and it will hatch and be a happy chicken without its parents, without ever meeting the parents. So you take away that effect of parenting. And what Per found was they basically stressed some chickens. And how they stressed them was basically by the chicken not knowing when it was going to get fed, to simplify it. So the chicken was a bit stressed in the way it ate. Okay, it was going around eating as quickly as it could in certain positions. Then when they took the eggs from those chickens and put them in an area with plenty of food, they showed the same stressed eating patterns, despite the fact that they had never been stressed. They'd always had plenty of food. They'd never seen their parents act like this, and yet they showed this stress effect. So they've inherited something in the behavior. Again, another great researcher here, Anita, also a young researcher at Linköping, just last year published a huge paper that was reported all over the world. And she's looking at flies. Flies are a fantastic model to work on. They're cheap, of course. No one gets too angry with you for working with flies. But they also reproduce really quickly. Now, in a human, you have to wait 100 years to look at 3 generations. In a fly, you have to wait 6 weeks. Okay? And what she found was when she took the fathers and she either starved them or gave them a really, really sugary diet, their children and their grandchildren were prone to obesity. But unlike a lot of other studies, she actually showed that this effect was actually dependent on some really important epigenetic enzymes. So this showed that this was going through epigenetics in some way. Anita now is trying to continue on to see if this effect can actually be detected in the sperms of humans. So she has a lot of student volunteers who are on a very sugary diet and then donating sperm every so often. And they're going to see if they can see an effect on the sperm that could possibly explain this in humans. So this would be one of those interesting studies where it could possibly be that your parents being very overweight does in fact affect your likelihood of being overweight regardless of how you're raised. But we're a long way from proving this in humans. But it's certainly a good functional step. And this is possibly the most well-publicized study of all epigenetic studies of inheritance so far. This poor little mouse was given something to smell from cherry blossom. And at the same time he was given something to smell, he was given a tiny little electric shock in his foot, just a little flick. But this was done over and over and over again. Then this father and mother had these kids. And whenever this smell was around, these mice got really jumpy. And these kids have never been, these pups had never been treated by this shock therapy. They had no reason to fear this effect, but they were very, very afraid of this smell. And even the grandkids of this original father showed this startled effect whenever this smell was around. And again, they went a little bit further in this study, and they showed that even in the sperm, there were slight changes on a particular receptor in the nose that detects this, okay, in these mice. But it has to be said, this is one of thousands of studies, and most of the other studies have failed to see anything. So we're again a long, long way from understanding is if our epigenetic changes can be passed to our children or not. So we're going to leave inheritance now and move on to, well, what about epigenetics in our diseases? Not the idea of passing epigenetic changes that affect the disease outcomes in our children. So epigenetics and disease. This is a very common picture to someone like me, probably uncommon to most of you. And what this shows here, these are all tumors, breast tumors, from hundreds of women. And each one of these rows, if you look very carefully, you'll see a row, is a gene. And if that gene is methylated, it's red, and if it's not methylated, it's blue. Okay? And what we see is, in all of these tumors, the methylation patterns are different. This is one of the reasons people got so excited about epigenetics and disease very early on. In cancer, one of the few universal features of all cancers is that your methylation is changed. So obviously people took this observation and the observation that we can change methylation and said, well, this could be a good target for therapy. And in cancer, again, without getting into the nuts and bolts too much, promoters are parts of your DNA just in front of genes. Okay, they're like the ignition switch for a gene. And in cancers, we see that a lot of these ignition switches are turned off by epigenetics. So that gene that should be turned on is now off because it's methylated. Okay? And often these are really important genes at stopping cancer from forming. Genes we call tumor suppressor genes. So by turning off all of these genes, the cancer cell gives it an increased ability to keep going, keep dividing, be more aggressive, be more aggressive. And again, these genes here, the TET genes, these are very important enzymes in our body. These are enzymes that can add and take away methylation from our DNA. And they are among the most mutated genes in all cancers, particularly in T-cell cancers, okay, in our blood cancers. So the fact that we see changes in methylation in cancers and we see mutations in these genes in cancers does suggest that this is a very important process. Epigenetics is important somehow to cancer. But in a study I did with a colleague in Edinburgh before I came here to Linköping, we actually found something quite disturbing. Found if you take a healthy breast cell, and you have two genes there, we'll say, and this gene is turned on and doing its stuff, and this gene is not on. A lot of genes that are not on are not methylated either. They're just not on. They're sleeping. We found that virtually all the genes that get methylated in the cancer weren't on to start with. So they're not being turned off by epigenetics. Epigenetics seems to be changing in the cancers, but we have yet to find a real functional role. How does it help make this cancer or push this cancer or make this cancer more aggressive? And this is a common problem in epigenetics, trying to differentiate between a consequence of the disease and a cause. Is the epigenetics changed because it's a cancer cell, or did the epigenetics make a change into a cancer cell? We come back to this incredibly important image published 15 years ago, showing increases in asthma, type 1 diabetes, multiple sclerosis, and this obviously is also observed in many other diseases, including asthma, eczema, and hay fever, allergy. Okay? Allergy, or hay fever in particular, is something I study in detail. Allergy has increased massively. One explanation for the increase in allergy is something called the hygiene hypothesis. And it states that when we're growing up, we're supposed to be crawling around in the dirt, getting bugs and bacteria on us all the time. And every time we do this, it trains our immune system to get tougher and tougher and tougher and tougher. But the cleaner the country you live in, Sweden being one of the cleanest, in which children are protected more and more from bugs and bacteria, the more allergy you have. The idea is that children aren't actually being in contact with enough bacteria and bugs when they're young, so when they get older, they're more sensitive to lots of things. A great example of this is, I just had a child 5 years ago in Britain, and we were told, don't let your child near peanuts in the first year, because it's so dangerous, because peanut allergy can be so dangerous. Whereas in Israel, all children grow up eating these little peanut snacks from age of 6 months. It's the first food they get. And Israel has the lowest levels of peanut allergy in the world. So after a huge study proved this was the case, 2 years ago, the NHS, which is the National Health Service in Britain, has changed the recommendation saying you should definitely give peanuts to your children as early as possible. So things are changing dramatically. So our early exposures to some things we call antigens, but these can be pollen from trees or peanuts or cat hair, helps prime and strengthen our immune system so we don't get allergies to these later in life. But it doesn't explain everything, okay? You take two identical twins that came from the same egg. They have exactly the same DNA sequence, exactly. They grow up in the same house, getting the same breast milk, going to the same school. these two will not get allergies to exactly the same things. Rarely ever happens. And if they do get allergies, they'll get them at different times. So it can't completely explain this. Okay? There is something to the hygiene hypothesis, but a hypothesis is just a proposal. And it's still called the hygiene hypothesis because it hasn't been proven yet. So what else could be explaining the rise in allergy? Could it be that some changes in our environment and the way we live are affecting our epigenetics in our immune system, making us more likely to have autoimmune diseases or allergies? And if we want to look at an autoimmune disease like diabetes or rheumatoid arthritis, you can view them in some ways as an allergy to yourself. Your immune system is recognizing parts of your own body as a threat, and you're getting inflammation in your joints or in multiple sclerosis in the nervous system. So as I said, we can't explain all of this by genetics or by the hygiene hypothesis. So could our altered environment be changing our epigenetics? Hay fever is a perfect example for an epigeneticist, a perfect disease to study. Why? It has very well-defined symptoms. Okay? You can tell when someone has hay fever. You can do little skin prick tests. They work perfectly. More importantly, you only have symptoms at a certain time of the year. So you can take the same person during the pollen season and outside the pollen season. It's the same person, has the same genes, and you can look at them with and without symptoms. You can't do that for most diseases. We know what causes it. It's pollen. Okay, so we can take cells from someone in the lab, put them in a dish, and add some pollen and see what happens to the cells. It's easy, get blood cells. Okay? It's easy to get people. People are happy to give you blood to work on. They're not so happy to give you bits of their brain or their liver. So it's an easy system to study. And we can do what, as I said, allergen challenge assays, where we take this blood cells from a person with allergy, and we actually put the pollen on the cells in the lab, and we can see at the molecular level how these cells are reacting to the pollen. And this is really important. There are lots of discordant monozygotic twins. These are twins that came from the same egg. They're genetically identical, but one has allergy and one doesn't. So they're a very, very good system to look at. Whatever the difference is there between those two, this is the difference that explains that extra proportion that's causing allergy that has not to do with the genetics. And what's really strange about allergy and hay fever is that you can cure it. You can actually cure it, and you cure it by giving someone something called specific immunotherapy. Specific immunotherapy is basically giving them pollen. You treat them with a little tiny dose of what they're allergic to over and over and over again, and suddenly they're no longer allergic, which in some ways supports the hygiene hypothesis in a little way. And Linköping's allergy clinic here is a leading allergy clinic in the world, trying and testing these new techniques and bringing them into fruition. A T cell. T cells are key to your immunity. They're controlling your body. They're little white blood cells, and they're going around your body all the time looking for threats. Is this just me? Is this a bacteria? Is this a parasite? And if it's something bad, they basically alert the whole immune system to say, kill this. That's what your T cells do. Without T cells, we're finished. The way HIV and AIDS works is by killing your T cells. When you have no T cells left, you have no immune system to defend yourself. You can die from a common cold. In the case of allergy, our T cells, for some reason, see pollen from grass or birch and say, okay, this is actually a parasite. Attack it. That's why you get these inflammation in your nose, these groggy feelings as your immune system starts working overtime to eradicate what it thinks is a threat to your health. Now, allergy for most people is a mild disease. There's a lot of over-the-counter drugs that work very well. But a lot of people who have allergy, hay fever, suffer enormously from it. And it really reduces their quality of life. Okay? So for some people, allergy is an incredibly serious disease, even here in Sweden. So understanding how it works and finding better treatments and seeing particularly if epigenetics is involved is very important research. I'm sure you've all seen family trees before, but this is just a family tree. Okay? But we're not looking at brothers, sisters, mothers, daughters. What we're looking here is the methylation of the entire genome of T cells from people who don't, who are not allergic in blue and people who are allergic in red. And when we do this and we say, well, how similar is the entire methylation in the genome of these people with allergy and without allergy, we find that most of the people who don't have it are very similar to each other, and most of the people who do have it are very similar to each other. So this showed us for the very first time, this research is done here in Linköping, that the epigenetics of people with patients with allergy is different from that from the people who don't have it, suggesting, okay, epigenetics is altered here. It doesn't tell us that it's causing disease, but it says that it has changed. And here's another statistical method that we use for separating, and it just shows you how clearly we can separate, with the exception of this one person here. There's one control that seems to have quite a allergy-like methylation or epigenetic pattern in their cells. Even more extraordinarily, we found that the symptoms of these patients during the pollen season was related very much to their methylation. Principal component 1 here, it's a very statistical term, is actually referring to the methylation in the cells. So we see in the patients, the methylations in their patterns in their cells predicted how bad their symptoms would be, which is an even stronger indication that the epigenetics might be playing an actual role in the clinical manifestation of the disease. But allergy is not the only common disease in which people have seen changes in methylation. Over the last 5 years, we have seen changes in the methylation in all of these diseases: diabetes, multiple sclerosis, asthma, lupus, eczema, celiac disease. And yet still, we don't fully understand whether or not these changes are a result of the disease occurring or are contributing to disease occurring. So what I and my colleagues working on epigenetics and complex disease will be doing for the next 10, 20, 30 years is trying to answer this question. Are we just looking at something that's happened, or are we looking at something that is causing the happening? But epigenetics is a field that has excited many people because of its possibilities of us controlling our own genetic destiny. But Adam Smith, the great economist from Scotland way back, I went to University of Edinburgh just like Adam Smith did. He said, science is a great antidote to the passion of enthusiasm and superstition. So don't believe what people are selling you about epigenetics. What we know at the moment about epigenetics is that it's important. We don't know how it's important. But we're going to continue researching on it and continue trying to understand and dissect its function in human health and disease so that eventually we can get to drugs for the treatment of these disorders. I hope I've been able to keep you all with me, and I hope I've maybe instilled a little bit of excitement in you for epigenetics, because whether you like it or not, you're going to start hearing this word a lot more in the popular news on Kunskapskanalen and on SVT Nyheter, because this is where all the research funding is going in medical science at the moment. Thank you very much for your attention. I'm happy to take questions in English or Swedish if you want.
Thank you very much.
Thank you.
Har vi några frågor här?
I've worked with transgeneration epigenetics, and I don't know. I saw that you left out some that you said that there is not much evidence for transgeneration epigenetics, epigenetic inheritance in the mouse model. So I want to know what is your reason to dismiss, let's say, the evidence in obesity or in reproductive diseases that are being transgenerationally transmitted?
There is some evidence. And I've worked on it myself with people like Emma Whitelaw in mice. And we do see some effects of intergenerational epigenetic inheritance in mice. But it's not directed like people would like us to. It's not like, well, you have this experience which affects the methylation of this gene, which leads to this phenotype. What we've seen most of all is that it maybe leads to some more variation or noise in the next generation, which maybe is what it does. It gives you a bit more plasticity in what your phenotype will be in the next generation. But to be honest, we keep referring back to the same studies in mice and in human. Now, I've been at this for 15 years, and we've seen an explosion in the research in this field, but we're still referring back to the same studies 15 years ago. as the gold standard studies, because there has been many studies really that have blown us away. You take this study in mouse in particular, for example. I mean, you're in the field, you know more about this transgenerational in epigenetics than I do, but you'll also know the amount of criticism it's had. But this is something, there's a lot of critics on both sides of epigenetics, a very emotive field. So I don't believe all the criticisms that come out either. But these studies have to be replicated in other mice by other labs, which has not happened yet. The same lab itself said they have replicated it, the same postdocs. But this experiment has not been replicated in another lab with scent and shock. No. Yeah, I try to interpret the results as, I'm being quite harsh on my interpretation of the results, I realize that, but I'm quite an old-fashioned scientist that way. I would like to see clear, reproducible evidence of something strong before I'm going to take it on board.
Några flera frågor, annars har jag en egen fråga här. Vilka läkemedelsstrategier tror du finns inom det här området, och vad kan vi förvänta oss i framtiden? Är det cancerbehandling som ligger först? Eller?
Ja, det är det. Ja, självklart, det är mycket i cancerbehandling nu. Det finns faktiskt precis nu över 50 olika clinical trials på gång av olika läkemedel som påverkar methylation och histone modifications, något annat jag inte pratat om här idag. Och det finns en som heter dicytabins som vi redan använder i många olika typer av leukemias, akut myelolytic leukemia och också i myelodysplastic syndrome. Och den här har de nu provat också i combination therapy i non-cancerous diseases också, men det har inte varit många jätteklart resultat från de här studier nu, men det finns så många på gång. Jag är jättehoppfull i alla fall att vi ska se några positive results i någon andra snart.
Är det mycket biverkningar? Är det riskabel behandling?
Ja, jag är ingen clinician, men dicytabin är oftast efter resistance. Så om den här primary behandlingen inte funkar, oftast kan du använda dicytabin. Det finns också nu mycket forskning på vitamin C. Också som det kan också förändras DNA-methylation också i cancer jättemycket. Så, och det är en säkert läkemedel. Du kan ta hur mycket som helst utan att göra något till din hälsa. Så de provar lite vitamin C också i akut lymphoblastic leukemias och andra sån cancertyper. Men we're a long way to go innan vi vet om det funkar.
Bra. Om jag får ställa en personlig fråga då. Dina kunskaper inom det här området, påverkar det ditt vanliga liv på något sätt? Har du några hypoteser som gör att du exponerar dina barn till exempel då för...
Nej, absolut inte.
Den här hygienhypotesen ska man
kanske en gång jag tyckte så, men det finns så många läskiga saker du läser om när du är en forskare att du kan inte bara tänka om dem hela tiden. Så nej, jag hoppas det i alla fall. Kanske, jag vet inte.
Du håller det objektivt.
Stackars barn, ja.
Du håller det objektivt.
Ja, jag hoppas det i alla fall, om vi kan.
Ja, jag tackar dig så väldigt mycket.
Tack så mycket,
Håkan. Thank you very much. Mycket bra presentation.